rabies virus. With this presentation, students will be able to understand the mechanisms of the rabies virus, recognize the importance of rabies awareness, and understand the cycle of infection by the rabies virus. Rabies virus belongs to the order mononegavirals, viruses with a non-segmented negative-stranded RNA genomes. Within this group, viruses with a distinct bullet shape are classified in the Rhabdoviridae family, which includes at least 3 genera of animal viruses, including the lyssavirus, which is the genus for the rabies virus. The rabies virus can affect a wide host of mammals, including but not limited to dogs, cats, rodents, and bats. The virus affects the nervous system, which includes the brain and the spinal cord. Incubation for the virus can take 3 to 12 weeks, with symptoms showing as early as 9 days or as late as 20 years. Rabies testing is not like testing for the flu. A fresh brain sample must be collected post-mortem in order to test for the rabies virus. The rabies virus is very relevant in this day and age. In the past 50 years, almost 25,000 cases have been reported positive in Texas. Just last year, 555 cases were reported in Dallas County, with 1 returning positive, and 181 cases were reported in Denton County, with 6 positive cases. Just this year in August, there were 2 bats that were reported to have come back positive for rabies in Dallas County. Vaccination post-exposure is 100% effective before symptoms start. And there are pre-exposure prophylactic vaccines available, though they are pretty expensive. The rabies genome encodes 5 proteins: nucleoprotein, phosphoprotein, matrix protein, glycoprotein, and polymerase. All rhabdoviruses have 2 major structural components: the helical ribonucleoprotein core, which can be called the RNP, and a surrounding envelope. In the RNP, genomic RNA is tightly encased by the nucleoprotein. 2 other viral proteins, the phosphoprotein and the large protein, which can be called ARP protein or polymerase, are associated with the RNP. The glycoprotein forms approximately 400 trimeric spikes, which are tightly arranged on the surface of the virus. The M protein is associated both with the envelope and the RNP and may be the central protein of the rhabdovirus assembly. Before modern control methods for rabies, it was common in the Western Hemisphere that many domestic dogs had rabies. And before European colonizers, rabies was likely only common in bats and skunks, and was rare or absent in domesticated dogs. It's estimated that from 1642 to 1782, that dog-derived rabies did not enter the Western Hemisphere. The introduction of the cosmopolitan dog rabies lyssavirus variant and the expanding dog population provided ideal conditions for canine rabies. The shift of the rabies in dogs shifted into foxes, coyotes, and other canines. This shift makes rabies much more of a threat to humans and made it that much harder to control. At the same time, bats affected by rabies posed a challenge in the elimination of rabies in humans and dogs. The first documented case of rabies in humans was in 1779, and in the early 1800s, the frequency and geographic distribution of rabies increased. 3 stages of rabies are recognized in dogs. The first stage is a 1 to 3 day period characterized by behavioral changes and is known as the prodromal stage. Animals previously friendly will become aggressive, and animals previously aggressive will become friendly. The second stage is the excitative stage, which lasts 3 to 4 days. This is the stage that is often known as furious rabies, due to the tendency of the affected animal to be hyperactive to external stimuli and bite at anything near. Animals in this stage also become hydrophobic. The third stage is a paralytic stage and is caused by damage to motor neurons by alteration of brain function. Incoordination is seen due to rear limb paralysis, and drooling and difficulty swallowing is caused by paralysis of facial and throat muscles. Death is usually caused by respiratory arrest.